Cancer is the biggest disease-related killer of Australian children, but a new initiative is hoping to change that, using genome sequencing to treat and prevent childhood cancers. Professor John Mattick is the Executive Director of the Garvan Institute and joins me now in the studio. John, thanks so much for dropping in today. This sounds extraordinary. Let's just first look at the rate of childhood cancer. The mortality rate has dropped, but there is still an overall increase in children getting cancer. Is that correct?
Look, my understanding is that there is a steady increase, but it's not terrible.
Okay.
You know, we've got larger population. There are other factors involved, but we've done very well in treating these kids for many years. You know, a generation ago, most kids who got cancer, particularly brain cancers, would die. About 80% are successfully treated by conventional therapies, but the problem with those is that the toxic effects of those therapies can damage them for a lifetime. You know, It's a violent treatment, and that's particularly bad in young people. And the other 20% that die, about 150 kids each year die from cancer, and that's a tragedy for their families. So
this genome sequencing sounds fascinating. You're essentially going to be taking tumours, I understand, from cancerous tumours, and how are you going to actually sequence that genome sequencing? How will it actually happen? In layman's terms.
It's a big word, genome sequencing. Well, genome just means all of the DNA in the cells. And,
yes.
And in cancer, we now know that there are mutations that happen in the genes that control our normal growth and development that cause the cells to go mad, and that's where tumours come from. They start to grow. You know. Now, over the last 5 or 10 years, as the cost of DNA sequencing has declined, and I should mention it cost $1 billion to do the first human genome, but we now do it for a couple of thousand, so it's come down a millionfold. What we've discovered, and we did this at Garvan in conjunction with other institutes around the world, was that cancer is caused by a range of mutations, but the spectrum of those mutations in our genes are similar in different tissues. So, for example, 15% of breast cancers are caused by a mutation in a gene called HER2, which causes 8% of colon cancers as well. So the drug that was developed to treat breast cancer can be repurposed to treat a subset of colon cancer patients, but you have to know that they're suffering that mutation.
Right.
So the way we can determine that is to sequence their cancers, to work out which are the mutations which are causing the problem. That then allows the treating physicians to select the right drugs, and often these drugs are on patent and quite expensive because they have to go through very expensive testing processes. So with kids, the project that the Lions Club International is supporting is to sequence the tumours biopsies from the kids who've got cancer to actually determine what mutation is driving their cancer. That immediately informs their treatment options and will give them much higher success rate. In fact, a paper was just published 2 weeks ago to show that the survival rates from this targeted treatment based on genome sequencing is 10 times higher than it is beforehand. So it's huge.
So this is essentially about being able to identify very, very early on exactly what kind of treatment a child might need and get onto it straight away.
And instead of hitting them with massive radiation, which can give them lifetime damage, we can perhaps use fit-for-purpose drugs that are targeted against their mutation. Also, the information will be fed into a database which we think will go global. So for those mutations which are new in the children, often young cancers are predisposed by inherited mutations in the family, so we can advise the family of that. So it feeds into research as well as immediately into treatment.
And John, do you see this also being applicable to adult cancers, not just childhood cancers?
Absolutely. Well, in fact, we're just doing what is now becoming more common in adult cancers. With adult cancers, the standard of care, I think, very quickly is going to be to sequence the tumour to try to determine what we call the driver mutations are. It's much more informative than just looking down a microscope at a pathology sample. It's still early days there, but the thing with kids is that they often don't have the resources, and the families don't have the resources to do this. So, in conjunction with the Children's Cancer Institute at Randwick and the Children's Hospital at Randwick and all across Australia, and our genome facilities at the Garvan, we're going to be able to provide that through this wonderful gift from Lions and the Lions community.
That's fantastic news, Professor John Mattick from the Garvan Institute. We so appreciate you dropping in today.
Pleasure.
Thanks so much.
Thank you.